vamos a tener al doctor Paul Szpak. Él está actualmente trabajando como profesor asistente de antropología en la Universidad de Trent. Él también preside el Centro de Investigación en Canadá de Arqueología Ambiental desde el 2016. Estudió su pregrado en la Universidad de McMaster y luego estudió su doctorado en la Universidad de Western University para posteriormente realizar un postdoctorado en la Universidad de British Columbia entre el 2013 y el 2016. Todo esto en un contexto siempre utilizando análisis de isótopos estables, los cuales se han concentrado principalmente en la región de los Andes, de Perú y de Chile, como también en el Ártico de Norteamérica, en particular Canadá, Alaska y Greenland. Se me olvidó cómo se dice en español. Lapsus. Pero bueno, dejamos entonces a Paul con ustedes. Y recuerden, por favor, que los que quieran escuchar la charla en español deben ir a interpretación y apretar la opción español. Adelante, please, Paul, thank you for joining us today. Let's start with this talk, this amazing talk about understanding camelid practices.
Okay, thank you very much, Fran, for this nice introduction and for inviting me to give this talk here today. I'm going to share my screen now. Okay, so I am going to talk about stable isotope research that I've been doing for the last number of years.
Estables que ya he estado haciendo durante muchos años en Andes, específicamente en Perú y Chile, utilizando el análisis de isótopos estables para entender cómo las personas y los camélidos interactúan en la época prehispánica o prehispana, con algunos casos de estudio. Antes de entrar entonces a los aspectos específicos, quisiera retroceder un poco y pensar cómo podemos usar los análisis de isótopos estables para hacer una contribución, un aporte significativo para el estudio de los restos animales en estos sitios arqueológicos. La zooarqueología tiene como objetivo, depende claro, del texto que uno consulta, pero hay una variedad de objetivos, pero más que todo queremos caracterizar las relaciones entre los humanos y otros animales a través del estudio de los restos. A través del análisis zooarqueológico, las personas han enfatizado distintos aspectos de esta relación. En estudios más tempranos, que siguen hasta hoy, muchas veces se concentra en la subsistencia, en la importancia económica de los animales. Luego, más recientemente, hay un énfasis también en la relación social, la importancia simbólica o social para...
Most recently, following what's called the animal turn in this developing area of human-animal studies, there's been an emphasis on what people have called relational approaches. So very explicitly trying to focus on the relationships between people and animals and their significance. Not so much in terms of what they symbolize about human society, but what they actually mean in terms of those real lived interactions between people and animals. So this relational approach seems very well suited to the goals of zooarchaeology. If what we want to understand are relationships between humans and animals, obviously this is a very good fit. In a lot of this literature, you'll see discussions of animal subjectivity and animal agency. And in many cases, I'm a little bit skeptical about actually connecting real methodological approaches with these sort of more explicitly theoretical pursuits. So my question is, how can we actually extract this information from zooarchaeological material in a way that is meaningful and is connected to reality? So I think this is where stable isotope analysis has a lot to offer, and specifically because stable isotope data provide us with actual information on the life history of animals. They tell us about how animals actually lived. So we can contrast this with the traditional kinds of information that we would get from studying animal bones at archaeological sites, which sort of species were represented, how were their bones discarded, how were their bones butchered, at what kind of context did they end up in. usually focused explicitly on subsistence and explicitly focused on the interactions that would be happening between people and an animal's carcass rather than between people and the living animal. So if we really want to understand the relationships between people and animals, this relational approach seems really very well suited to using stable isotope analysis and similar lines of evidence to actually help us to understand these relationships in a meaningful way between the living animal and the people, rather than between simply the animal carcass and the people. I think we can get the best insights with respect to this kind of approach from focusing primarily on domestic animals too, rather than on wild animals that might have been hunted. So how we think about these relationships, I think, is very significant, and there's a big difference, in my opinion, between how herders, people keeping livestock, would have interacted with their animals versus how hunters might have interacted with these animals. So an earlier model published in the 1980s by Tim Ingold contrasted relationships among hunters and the animals that they hunted as one that's based on trust and mutual respect, with relationships between herders and their animals as one that's based on domination. And Ingold explicitly links the human-animal relationships in agricultural settings to sort of a slave-master kind of relationship. About 10 years ago, John Knight, who's a social anthropologist, criticized Ingold's approach and said that really the opposite is likely true, and that herders have a much better opportunity to get to know their animals in particular, whereas hunters can only know their animals in general. So hunters have very short and non-repeating interactions with individual animals, which means that they can get to know an animal type, a species of animal, for example, very well, but they never really get to know an animal as an individual. Whereas herders have regular interactions with particular animals that they might keep over many weeks, months, years, and that allows them to know those animals very well in particular, having relationships that are therefore more personalized. So if we can diagram this up, we have herders on the left and hunters on the right, and we can see that the interactions between the hunter and the deer that this hunter is hunting are short, are not especially meaningful at an individual level, though the hunter knows quite a lot about deer based on these repeated interactions. The herder, on the other hand, knows about whatever kind of animal that individual keeps, sheep, goats, camelids in this case. He knows them very well in general, but also in particular because of having these repeated interactions over and over again. So stable isotope analysis, I think, actually allows us to start to look at these kinds of relationships, what their nature might have been in the past, by allowing us to gain insight into how the animals actually lived, because that's what we want to learn about, the lives of the animals, rather than simply the circumstances surrounding the death of the animal and what happened to that animal's carcass after it entered the archaeological record. So I won't talk much about the underlying principles of stable isotope analysis, but I'm focusing on stable isotopes of carbon and nitrogen today, and particularly those isotopes as we study them in organic tissues in the body. These are very useful for studying the diet of past human and animal populations. And with respect to herbivores, we're talking about camelids here today, these stable isotope values change because of differences in the diet that the animals might have had, as well as differences in the environment in which those animals may have lived. And some of the important environmental variables that might cause isotope values to be different include things like variation in rainfall, local temperatures, the kinds of nutrients that were accessible within the soil. We also have distinctions between wild and agricultural plants in some areas. So where we have extreme environments, such as we get in some of the coastal areas in the Andes, the conditions might be extremely dry, but agricultural plants, which might be fed with irrigation, are buffered against some of those effects. So when I initially started to look at stable isotopes in camelids, this is a really ideal sort of setting in the Andes to study these kinds of interactions because of the great environmental diversity, both naturally as well as based on the kinds of crops that people would grow in these different regions. We have very big differences going from the coast at low elevation up into the highlands with respect to things like temperature, rainfall, and the kind of plants that would be available for animals to eat. So by looking at the sorts of isotope values we see in plants in these different areas, we can make some predictions about what the isotope values would be for camelids living in a number of different conditions. So for example, we get much higher carbon isotope values, which we see here on our x-axis in C4 plants, things like maize in particular, than we do in C3 plants, the majority of other plants we'd see in the area and the other crops that we'd see in the area. Dry conditions cause us to have higher nitrogen isotope values, and in some cases, for some plants, higher carbon isotope values. So this gives us a basis with which to be able to interpret what's going on in our herbivores. So the initial purpose of my work with camelids was really pretty narrowly focused on answering this question of whether or not these animals were restricted to just high altitude habitats. So were camelids actually being raised outside of the high altitude grasslands of Peru is where I focused my research initially, using stable isotopes. So what we did was we used baseline data from plants, the isotope values from plants in the region, to generate some expectations for what animals living under different conditions would look like in terms of their isotope values. And we looked at bone and hair from camelid remains, as well as fiber from archaeological textiles. So these animals that do live in the highlands, they have a certain set of characteristics in terms of their isotope values that should make this kind of pattern of animal husbandry, keeping animals in these high altitude grasslands, fairly distinct. Particularly, they've got lower carbon isotope values, which means they're eating mostly C3 plants. So they seem to have most of the time less than about 20% C4 plants in their diet. And they've also got relatively low nitrogen isotope values. Although this can vary, we would expect this to vary quite a bit depending on what the conditions are in the local high altitude grasslands. So we don't have a lot of data that speak to this, but I would expect, for example, big differences in the isotope values, or at least subtle differences in the isotope values between camelids living in the Bofedales, the dry puna, and the salt puna, for example. We generally see for these highland animals a low amount of variation among individuals. So the isotope values of one individual tend to look pretty similar to another individual from a certain site or a certain region. And we've seen this kind of data and this pattern with bone collagen, so the protein part of the bone from camelids that we've analyzed from a Raquai site called Chinchawas in the Cailan de Huaylas, as well as at Tiwanaku. We generally don't have a lot of data from highland animals, typically because the preservation there tends to be not as conducive to these sorts of analyses as is the case on the coast. So the first study, case study I want to talk about is with material from two sites in the Viru Valley, which some people might be familiar with as the area where Gordon Willey did his sort of landmark settlement pattern studies in the 1940s. And so these are primarily early intermediate period occupations between about 200 BCE and 600 of the Common Era. The first site is Huacagainazo, which was the capital of an EIP polity known as Viru, previously often called the Gainazo polity. And then another site called Huaca Santa Clara, which was a regional administrative center also associated with this Viru polity. Camelid remains are very, very common at both sites, and they are by far the most common type of vertebrate that we get at these sites. But that doesn't necessarily mean that these animals were living in the area. So were they actually raised locally, or were they imported from highland environments, either as meat on the hoof, sort of living animals, or maybe dried meat in the forms of charqui, for example. So Huaca Santa Clara, this regional administrative site, is also interesting because after the site was abandoned at the end of the EIP, it was reused for a ritual sacrifice that involved humans and juvenile camelids, and this was around 1100 of the Common Era. There's very good organic preservation of this ritual event, lots of hair and other soft tissues, for example. And this gives us an opportunity to look at whether or not there might have been differences over time. So between the early intermediate period and sort of the terminal middle horizon, very beginning of the late intermediate period, as well as under different contexts. So more domestic contexts versus ritual contexts. So we've got a number of different camelid remains from 4 different periods at these 2 sites, a fairly large number of individuals. And the isotope values look something like this, which probably doesn't mean a whole lot. But once we compare them directly to one another, we're going to be doing that with these ellipses, which are these shaded areas that I have for each of the 4 panels. And the ellipses are just a nice way of condensing this bivariate data down into one sort of cluster that tells us about the amount of variation, the central tendency, as well as any correlations that might exist between the 2 variables. So we compare these 4 sites to one another, as well as to animals that we know live in the highlands. So that is the black ellipse here. And what we can see is that, generally speaking, these 4 groups are very distinct from animals that did live in the highlands. And specifically, they've got diets that have a lot more maize in them, or some other C4 plant, possibly grama salata, but I think more likely maize. And they also have a lot more variation. So the size of these ellipses tends to be much bigger. That's what's being shown in the right-hand panel here is how much variation there is in each of these groups. Much more variation in these groups than there are in the animals from the highlands. The 4 groups here at the different periods at these 2 sites, though, are quite similar to one another, much more similar to one another than they are to animals from the highlands. Generally speaking, these data then are inconsistent with these animals being imported from the highlands, or if these animals were imported from the highlands, they were being raised in a very different way relative to what we see with other highland archaeological sites or with modern animals that live in these high altitude environments today. So this definitely suggests that we have localized camelid husbandry at low altitudes during the early intermediate period. What this husbandry might have looked like, I think, is that animals would have been fed crops, particularly maize, as well as some others, but were not eating wild coastal plants. So in these areas, the coastal wild plant productivity would have been fairly low, and much of the good land would have been used for agriculture. So this does make some sense. The other thing that makes this more likely is if we had animals eating wild coastal plants, we would expect them to show much higher nitrogen isotope values, which is characteristic of very arid environments. And that's certainly what we see in modern plants, wild plants, growing in these coastal locations. One problem that we always run into with stable isotope analysis, though, is the problem of equivocality, or multiple interpretations being possible to explain any one set of data. So do we see really diverse diets in these individuals because, really diverse isotopic variation in these individuals because they had diverse diets, or because they came from a lot of different places? Both of those explanations could account for the data that we see, because you can control an animal's diet regardless of where you raise it and sort of get the isotopic values to look like this. So I think where my opinions are much more in line with its diverse diets rather than diverse origins is when we look at the sacrificial context. So that ritual event at Huaca Santa Clara, which postdates the abandonment of the site by about 500 years. So these animals are all very young. Most of them are less than 3 months of age, and all of them are less than 9 months of age. And this sort of long distance travel is fairly difficult with animals that are this young. And interestingly, we see a very similar range of variation in the dark red circle, which is our late sacrifices, relative to the earlier occupations at the site. So we see a similar sort of isotopic range, as well as amount of isotopic variation in these sacrifices as we do at the earlier periods at which this site was occupied. So we have to think then that either it's a pretty big coincidence that people were getting animals from a diverse range of geographic areas in the early intermediate period. The site was abandoned and people came back 500 years later and happened to get animals representing the same range of geographic areas. Or it's possibly the case that people raised their animals in this sort of diverse way, fed them a diverse range of diets during the early intermediate period, and those practices continued through the abandonment of this site and into the terminal middle horizon and earliest part of the late intermediate period. So my interpretation of these data is that they're more consistent with diverse diets rather than geographic origins being diverse. So a question then is, why so much isotopic variation? I guess if we're willing to accept that my interpretation might be correct, that we get a lot of isotopic variation because there was a lot of variation in the diet, why would that be the case? The data that we're seeing here are from the collagen in bone, the protein part of the bone. And the isotopic signature that's represented from this part of the bone is a very long-term picture of the animal's diet. So this is representing diet over likely at least the animal's entire life for those sacrifices, which are very young, and for older individuals, a very significant portion of their life, likely several years at least. So this means that any differences that we see in the diet among individuals are evidence of sustained long-term variation in diet. So it's not sort of a short-term sampling error. If we have two individuals, say on our top left-hand panel here, that have these sort of low values and these high values, those two individuals lived very differently throughout the course of their life. So what I think this is characteristic of is that people were keeping really small groups of animals that were fed distinctive diets from one another in these coastal river valleys. And that there was no particularly standardized diet that these camelids were fed. So people were essentially feeding them whatever kinds of crops they had access to, which varied pretty strongly among different households or other social units in the area. So what we didn't have, I think, are very large herds of individual camelids living together, because under those circumstances, I would expect to see less isotopic variation. You have one large group of animals, they should tend to resemble one another in their diet, compared to a situation where you had many small groups of individuals, each being managed by different people, and those different people making distinct choices, whether because of what was available or because of certain opinions that they had on what was the best way to raise one's llama or alpaca, causing these large scale variations in the diet. This might also be a pattern that's sort of forced by the more urban conditions in some of these larger sites, where we have people and llamas living together in very close proximity to one another, which would be very different than what we might see in these high altitude grasslands. An important question that we have, if we accept the premise that these animals were living locally in these areas, is how integrated was the raising of livestock and the growing of crops? So is it the case that the animals were fed the crops, the animals produced fertilizers that were then used on the crops, which then fed to the animals, and so on? I think this is likely, but right now we don't have a lot of data to support this. So I think we have good evidence from this first case study of animals being raised outside of the highlands. It suggests that herds in these coastal valleys as early as the EIP were raised locally. And some predictions that we could make that would support this hypothesis would be, if we had other stable isotope markers, they should be consistent with this. So if we could look at markers that told us about explicitly the geographic origins of these animals, we should see much less variation in those and much more variation in the dietary markers that I've just talked about. This is something that I'm definitely interested in doing in the near future. We should see this pattern maybe at other sites in the region. And we should see evidence that camelids had, or camelid meat in particular, had an important role in the local human diet. And possibly the evidence for the use of camelid dung as fertilizer. If you have these animals present, it stands to reason that you might make use of their dung to increase the productivity of your crops. So related to one of these, this is some work that one of my students did with human remains from these two sites, Huaca Gallinazo and Huaca Santa Clara. And we're comparing them here to plants growing in the region as well as camelids from these sites. So the camelids are the black crosses here, and then all the dots represent isotopic data that's corrected for discrimination according to trophic level, so we can directly compare these values between the foods and the animals that were eating the foods. And what we can see is the humans here have isotope values that are very much intermediate, primarily between the camelids themselves and the C4 crops. And so this is indicative of those two resources being the primary part of the diet. So this does support the idea that camelid meat would have been important in the area, in addition to the fact that camelids were by far the most common animal bones recovered from these two sites. And now what about other data from the region, from other parts of the North Coast? So this is a summary plot that Elise Dufour made comparing data from a number of different sites, including the ones that I just talked about. And generally speaking, we see similar, although subtly different patterns at other places on the North Coast. So other sites in the Viru Valley, sites in the Moche Valley, in the Jequetepeque Valley. And in some cases, we see, for example, here, this red circle at Huanchaquito, quite a lot of maize, much more maize and consistently more maize in the diets of those individuals. So in some areas, we get a fairly similar pattern, but in other areas, we get something quite different. In none of these sites, though, do we get something that looks like those high altitude camelids that I showed you previously from Chinchawas or from Tiwanaku, which suggests that these data are much more consistent with people from different areas of the North Coast raising animals locally and doing so in sort of a localized way. So all these values distinct from the highlands. So the next set of data I'd like to talk about are from some sites in the Santa Valley, also on the North Coast. And here we've got, again, isotopic analyses from two sites, El Castillo, which is from Moche 3, and Guadalupito from Moche 4. And here we've got isotopic data from bone collagen, again, which also shows a huge amount of variation among individuals. So very, very similar to the patterns that we saw in Viru, suggesting overall higher amounts of C4 plants in the diet. But again, lots of variation among individuals, and actually even more variation here than what we see in Viru. This suggests that we had fairly similar animal management practices going on here relative to what we saw at Huaca Santa Clara and Huaca Gallinazo. But an interesting thing we can add here is to look at isotopic analyses of textiles made from camelid fiber, in addition to the analyses of the animal bones themselves. So we can use the data from our bone collagen. Our group from Chinchawas is, I think, our best example of what animals raised in the highlands might have looked like. And then if we sort of accept my hypothesis that the animals from Viru were local to the coast, that's what we might see from, say, coastal animals. And then we could compare these to different data sets for textiles. So the first of these that I'll talk about is not actually from the Santa Valley, but is from a private collection that was donated to the Canadian Museum of History. And this is associated with the Chancay on the central coast. These are really, really fine textiles, and they don't have very good contextual information because they were in this private collection, I'm sure from looted materials from earlier in the 20th century. But when we look at their isotope data, they're this black area here. They're much more similar to the highland animals we have from Chinchawas than we do from the values from the animals from Viru. So these data are very consistent with our expectation that while we might have had some local animals living on the coast, probably for meat, for textiles, particularly the kinds of fine textiles we see in the Chancay area, that seems to be coming from imported fiber from the highlands, which is what we would expect. Now, if we look at the textiles from Viru, from Huaca Santa Clara and from Huaca Gallinazo, we see something very different than what we see with the material from Chancay. We see values that once we correct them to make them directly comparable between bone and hair, they look very similar to the data from the bone collagen from Viru. And this suggests that these data are much more consistent with the camelids from Viru and the textiles, the fiber from the textiles coming from animals that were sort of the same population, essentially. So we either have a similar origin for the material that ends up in the textiles and for the animals that end up butchered at the site, or we have a very similar sort of pattern of animal management for these two different groups. This suggests that maybe the textiles were actually produced from local animals in Viru, which is surprising because while it seems plausible that, that llamas may have been local to the coast, it seems much less likely that that would be true for alpacas, which are apparently far harder to raise in a diverse range of environmental conditions. When we look at the data from the Santa Valley, we see the same thing as what we saw in Viru. So on the left, we have our data from bone collagen. On the right, our data from the fiber from the textiles. Very, very similar patterns of variation in both of these two different sets of data, again, suggesting that we have local camelid husbandry, not only through the production of camelid meat, but also for the production of fiber. So this is a pretty surprising finding and not actually what I would have expected, where we have data that are consistent with local animals being used for fiber production. I definitely had thought that we would see something much more similar to what we saw for Chiang Kai, where those data are consistent with the fiber being imported from the highlands. And one last set of data we have is for Pakatnamu in the Heke-Tepeke Valley. Again, we see the same kind of pattern, lots of variation isotopically, overall higher amount of C4 plants in the diet, very comparable to what we see in Viru and in Santa. Interesting thing about the Pakatnamu textiles, as well as to some extent Santa and Viru, is the quality of those textiles is much poorer compared to what we see at Chiang Kai, which is a very fine type of textile. So it might be the case that in some situations, people were more reliant on imported fiber to make really high quality textiles, but in other situations, we're more reliant on local fiber production, maybe to make primarily more utilitarian works. So in general, I think we either have a very coincidental situation and a diverse range of geographic origins for animals that end up at all of these different sites in all of these different areas, or we might have a form of camelid husbandry on the North Coast that was fairly widespread and fairly common. Again, because of this problem of equivocality or multiple interpretations, it's very hard to know for sure, but I think the increasing amount of evidence all tending to look fairly similar, I think points to the issue of small-scale localized husbandry on the coast being more likely. So what we can do is summarize all of these different data based on these two sort of important variables, which is the amount of C4 plants in the diet and how much variation we see among individuals. So our amount of C4 plants in the diet is on the y-axis, and our amount of variation among individuals is on the x-axis. And the blue points here down in the bottom left are the data that I think we have good reason to believe are from highland animals. And the red are those that I think are more consistent with this sort of coastal mode of husbandry. And since I put this graph together a number of years ago, there's been multiple other studies that have produced similar data with sites, different sites in the coastal region that tend to look more like these red dots than these blue dots. So this is just another way of representing that same kind of information. So in northern Peru, I do think we have this mode of small-scale camelid husbandry on the coast. The fiber that's used in textiles seems to have been imported on the central coast, the Chiang Kai data that we have, but these are very, very limited data. It's not a great ideal collection to study. So I would be very hesitant to say anything conclusive about this. But on the North Coast, where we have much better data from the textiles, from materials actually found in situ at archaeological sites with good context, I think we can be a bit more confident that there was probably localized production of fiber in the coastal valleys, not just in the highlands. So for my third case study, I want to move a bit further south into northern Chile, which has some very important differences between northern Peru and northern Chile. First of all, we have much more arid conditions in the high altitude grasslands in Chile relative to in this northern part of Peru that I've been talking about. And the valleys, the northern valleys in Chile are much more marginal in terms of agricultural productivity. So when I initially started to work on this with Daniela Valenzuela, I was very skeptical that we would see any evidence of localized camelid husbandry here. I thought whatever we would see would really be indicative of exclusively highland origins for these animals. We have also a limited number of comparative data for this region relative to what we have for northern Peru, although that was the case in northern Peru 10 years ago. There was very limited data, so we have to start somewhere. And finally, we don't have a lot of data from plants from this region to compare our animal data to. So the material I'll talk about here is bones from late intermediate period and Inca sites in the Utah Valley and textiles from late intermediate period and Inca sites in the Utah and Camerones valleys, the sites between about 1,000 and 1,500 meters elevation. So not right on the coast, certainly, but definitely in these northern valleys of northern Chile. So the data we have for the bone collagen first, we have very different patterns from the late intermediate period in our blue here with the squares and the late horizon, our Inca period here with the red circles, which suggests that there was either different geographic origins or different management practices for these camelids, or both potentially. Again, with our many interpretations being possible, it's hard to identify one as particularly likely in this situation. What we do see also is a lot more variation in the late horizon relative to the late intermediate period, which suggests that maybe the Inca had increased the different areas from which camelids were being sourced, at least for meat, because these are data that we see for bones rather than for textiles. We compare these data from Utah in the LIP and late horizon to other sites. So Tulan we have kind of in this area. We tend to see these data being more similar to what we get in high altitude areas. So we have some data from Tiwanaku, for example, which has lower carbon isotope values than that is comparable to many of these data from Utah. But we don't have a lot of data here, so I'm not very, very confident about this interpretation. And I would say the geographic origin is relatively uncertain at this point. And what we really need is some more data from archaeological camelids from the highlands of northern Chile to be sure. But I think these are data that are not consistent with localized husbandry, but that'll become a little bit clearer when I talk about the textiles from this area. So when we look at the textiles from these sites, we see something quite different. We see a lot more variation isotopically than what we saw for the bone collagen. This might just be an effect of sample size because we have a lot more textile samples than we did for bone. But what's interesting here is that we have 3 samples of human hair from a site in the Camerones Valley and 1 from a fox in the area. And we get these 2 distinct groups of textile samples. This one here, which I'm saying is, I'll say, probable highland origin, and this one here, which seems to be not likely to be of highland origin. There's no regional in terms of the different valleys or temporal organization to these different groups. The groups really do seem to be equally distributed across valleys and across time. And interestingly, when we look at the human data, which we think are probably people that lived locally in the valley, they're very similar to this second group of textiles that I'm saying seems to be from non-highland individuals. They have much higher carbon isotope values and much higher nitrogen isotope values than this other group here, which is what we would expect to see for animals living in a very arid region like this. So our first group, when we compare these to the bone collagen, our first textile group, which has about 85% of the textiles, looks very similar isotopically to our 2 bone collagen data sets. Our second textile group is very, very different though, and that's the one that contains our human hair samples. So I'm suggesting, or we are suggesting, Daniela and I, based on this, that there were some animals kept locally in the northern valleys of Chile that were particularly used to produce wool. It might be the case that there were also animals kept locally for the production of meat, but we just don't have enough data to see that yet. But so far, it seems like the bones are more consistent with being from high altitude habitats, textiles mostly from high altitude habitats, but some probably local. And these local individuals had diets that were very, very heavy in maize or possibly grama salata or some other kind of C4 plant, C4 grass that was growing in a very arid environment. If we compare that C4 component of the diet for our two textile groups here in purple to some other sort of animal groups, we can see that we have highland animals here in green. These are the bone collagen data from the Utah Valley, and then our first textile group, which I think is from a highland group of animals, sorry. And those all have low C4 content in the diet. Our second textile group, very, very high C4 dietary content of about 80%, only comparable to this very unusual group of llamas at Conchopata, a Wari site where there seems to be a very intensive maize provisioning. So these animals must have been fed a very restricted diet, either in an area with abundant C4 grasses or where maize was very much predominant and fed specifically to these animals. If we compare it to what we see on the north coast of Peru, for the sites I've talked about before, we get more of like a 50-50 C4 to C3 contribution, whereas with our unusual textile group from northern Chile, we get a much higher contribution of C4 plants. So I think what we have in northern Chile is limited evidence for local camelid husbandry in these northern valleys, which is distinct from what we see in Peru, where I think we see much more widespread localized camelid husbandry. We see some kind of difference between the late intermediate period and the late horizon in the bones, but not in the textiles, which I think is very interesting, and I do not have a good explanation for. I would have expected if the Inca were going to influence any sort of supply chains, it would be related to textiles rather than to animals being used for meat, but that seems not to be the case. Again, this might simply be an artifact of sample size, and if we had many more samples of bone, we would see a different pattern. It's hard to say. What we definitely need is more camelid data, especially from the highlands, to be more confident in saying that this first group of textiles and these bones are actually from highland animals. Right now, we don't have a very good comparator group like we did for northern Peru, and that's a very critical area that we need to work on. So to summarize this stuff briefly now, the stable isotope data, I think, reveal a much more complex nature of human-animal relationships that existed prior to the arrival of the Spanish. I think we have good evidence for herds of animals being kept outside of the high-altitude pastures. I think that at this point is not a controversial statement to make. Camelids seem to have been kept in a really diverse range of environments, both for their production of meat and for the production of fiber. I'm not 100% confident that this means people were keeping alpacas in the coastal river valleys of northern Peru or of northern Chile. We don't have data to suggest that specifically, but I think it's a possibility. Or it's a possibility that there was some sort of fine fibered llama that might have been used in the past that we don't have much evidence of existing past the conquest. So we really need more data that are explicitly linked to geographic origin. So here I'm talking about oxygen isotopes and strontium isotopes to add to our carbon and nitrogen isotope data to strengthen those interpretations that I've talked about. Some future questions that I think are going to be important is how widespread was this camelid husbandry in the coastal valleys? We've got a fair amount of data now, and certainly much more than we did 10 years ago, but there's still a lot more work to be done. When did people begin keeping these animals in these coastal river valleys? Does it date back to the early horizon? Is it much earlier than that? Was there experimentation at different points in time in these different valleys? We really don't know this at all. Were the animals kept for meat, for fiber, or for both? And how did this vary among areas? So again, we don't really have good comparative data for this now, but it would be really interesting to get more of this. And something I'm especially interested right now in is, was dung from the camelids being used as a source of fertilizer in many of these coastal valleys? So at sites where we have what I think is good evidence of people keeping animals locally, were they also using the animals as a source of fertilizer to improve the productivity of their crops? So thank you for listening to that. And thanks again, Fran, for inviting me to give this talk here today.
Thank you very much, Paul. That was very nice. ¿Hay alguna pregunta para Paul, por favor, por el chat? Ok, hay dos preguntas y están en inglés. So these questions are in English. So I'm going to read them in English. Nice talk, Paul. So what could explain the enrichment seen in both nitrogen and carbon isotopes from the oldest to youngest camelids at the virus sites, climate change, shift in diet? And the other question is, what does husbandry mean in this context?
Okay, first I can see the chat now. So for Claudio's question, I think that difference is probably related to the age, not the geological age, but the age of the animals. So the sacrifices are from a very restricted age group and very young animals in particular. So I think the nitrogen isotope values are higher there because we have a much stronger nursing signature than we do in the other group of animals, which has a mix of ages from young animals to very old animals. And I think we also might just have a bit of a sample size issue there, that we've got less variation and a bit shifted in the sacrifices, largely because it's 25 animals that were born at the exact same time and died at the exact same time. So our other sites, we've got 150 or 250 years of occupation with camelids associated. So I think we might just have a bit more variation there.
Mm-hmm. And
the other question.
Yeah, what does husbandry mean in this context?
In terms of what am I using the word husbandry to refer to? Do you think that means?
Daniela, sí? Sí, yes.
Okay. Yeah, so I'm talking about the range of interactions that exist between people and animals, how they care for their animals. So there's a number of other things that I think we can't see evidence of with these kinds of data. But the dietary part, I think we're fairly well positioned to actually see evidence of husbandry as it relates to diet. So there may be various other aspects of these interactions related to how people might have controlled breeding, how they moved across the landscape to some extent, that we can't see good evidence of. So I'm just focusing mostly on the diet because that's what the data actually allows me to speak about, I think.
Mm-hmm. There is another question. It says, excellent talk, Paul. I'm curious about your thoughts on inter and intra-site comparisons. Do you think that the isotope results could be interpreted differently if a spatial control within each site is considered?
Can I ask what's a spatial control to Alexa?
The context where the samples were found.
So you mean if there was, I guess I'm not really clear on the question, to be honest.
Elite versus non-elite.
I think that's possible. I think, yeah, that would be interesting. We have some ability to look at that in the Santa Valley, but the problem usually is that I think we get smaller and smaller sample sizes when we try to divide them up into finer and finer units of elite, non-elite, domestic, administrative. In Santa, I can say there's no difference, but in that situation, the sample size is also not enough where I'm very confident in that. But I think it's entirely possible that that difference could resolve itself if we were able to chop the data up in that way.
Okay. Here we have a question in Spanish, so I guess Pedro will translate it for you. So, se ha enfatizado en los restos óseos y las fibras de los camélidos para la identificación de colágeno. ¿Se puede utilizar los coprolitos para el mismo propósito?
I am skeptical about using the coprolites. So with collagen and with the fiber, we have a high degree of confidence about what's in those samples. So we know how much of various elements there should be, how much carbon there should be, how much nitrogen there should be, not related to the isotope values, but that gives us a very good idea about how the material might have been altered once it entered the archaeological record. I think with the coprolites, that's much harder to know. And even short-term variations can cause big changes. So particularly if we're composting, say, the dung, that can cause really big changes in the isotope values, even over a few weeks or a few months. So I'm much less confident about taking data from the coprolites and saying that tells us a lot about the diet of the animal, because we're not sure about what changes happened to the coprolites in terms of their isotope values between when the animal deposited the coprolites and then eventually when we found them, as the processes happened in the archaeological record.
Okay, so there's a question of Ramiro. How do the results compare with France regarding the importance of coastal camelids?
Is this Fran's results with Elise and Nicolas, or Fran's results in Chile?
Ramiro, Chile. Sorry, it's not published yet.
I guess the only thing I could talk about is the textiles from the radiocarbon paper, maybe.
Mm-hmm.
Yeah, I think those are really interesting. Those data are more interesting than any data that I have. And I think, yeah, I'll let Fran talk about them. It's her data.
Well, I think it is a bit different in Tarapacá to what you presented in Arica. For some reason, the values are higher for carbon and nitrogen in the coast of Tarapacá, and also in the oasis of Pica, as you know. We have some more results now, and I'm really sorry I haven't published them yet, but it will happen soon. But yeah, I think it's likely related to the fertilizers and the ingestion of fertilized crops.
Yeah. I hope so.
Yeah. Okay, so we have another question in Spanish. ¿Qué indicadores están considerando para hablar del uso de guano como abono? ¿O cómo pueden decir que efectivamente se usó el guano?
Do we mean guano specifically from seabirds, or this is guano in terms of any kind of animal dung?
Mariana? Dung.
Yeah, I think there are some data from some of these sites that have very high nitrogen isotope values, not high like Fran has, but high in the context of northern Peru, that might be indicative of the use of dung as a fertilizer. I think the best data that we could get would be from isotopic analyses of the crops themselves, and large numbers of these crops from these same sites where we have camelids. And particularly if we had sites where we don't seem to have the presence of camelids, and those where we do seem to have the presence of local camelids, how do the crop values compare there? But we're not at that stage yet where we can answer those questions.
Mm-hmm. Okay, so there is a question of Mai Takigami. Is there a possibility that they made textiles by mixing camelid wool of different origins, for example, rewind and reuse the threads? Yes,
I think that's very possible. And I also think this is why this technique can be good for trying to assess the origin of fibers when you look at a large number of samples. An individual sample, I think, is very hard to interpret, because you're right, it could be multiple animals. It could be multiple animals from multiple different geographic origins. It's very hard to know. So I think the only way to make any kinds of statements, again, tentative as they might be, is to have larger numbers of samples to characterize the overall pattern of variation.
Mm-hmm, mm-hmm. Okay, and there is a last question from Ramiro. Do you think strontium isotopes will be promising there in Peru?
Yes, I think so. So my student whose data that I showed, the carbon nitrogen isotope data, she also has strontium isotope data from those humans that are associated with the sacrificial camelids. And there is absolutely at least one individual that's from the highlands. And then there's two individuals, I don't want to give away all of our results too much because it's also not published, but there's two individuals that have highland style cranial modification, that have very local strontium isotope signatures in the teeth. So I think the relationships between the coast and the highlands are probably also more complicated than we know. We have multi-collectors at Trent, and I think it would be very interesting to do some work with these particular camelids too, to put my own hypotheses to the test. So I think it's promising, and I think I will eventually get around to that, or at least a student working with me will get around to that.
Mm-hmm. Okay, right. That's it. We don't have more questions, but thank you so much, Paul, for this very, very nice talk. And thanks everyone for coming to the seminar. Muchas gracias a todos y todas. Los esperamos en 2 semanas más para la charla de Irene Lantos sobre residuos orgánicos en cerámicas. Gracias nuevamente y nos vemos pronto. Adiós. And bye, Paul. Thank you so much, Paul.